So, welcome everybody, and first of all, thanks to the organizers for having me here at the iPoLS meeting. I will continue to talk about cell migration in the particular case of cells being confined in surface geometries, and I will give you essentially a few examples what this might be useful for to understand cell migration better. I'd like to start with a statement that I find very wise. It says, living cells are the test tubes of the 21st century. Now, you could interpret this in many ways, but what I think of it is that we have technology at hand that we can measure inside single cells the smallest possible unit to do experiments. And we like to do experiments in a quantitative way to understand reaction kinetics inside living cells. So in that sense, I will give you a few examples on single cell response functions that we'd like to measure in an automated way using fluorescent reporters. And in order to do that efficiently, we pay attention on how to seed cells. We like to array them on sites such that they are nicely accessible. And in order to do this, we need some kind of surface patterning or surface chemistry that we introduce. The students that are doing this work, I'd like to acknowledge right from the beginning. In particular, Caroline and Peter Rutgermann's work I will show. Sonja Westermeier is also here in the audience. And as you will see then, cells that sit on pattern can have a particular behavior, and one of the behavior is that the motility is confined. And in the second part of the talk, I will give you a very nice example of collective behavior on small islands. This is work by Felix Segeras, who is also sitting somewhere here in the audience, and as well as Matthias Swan. So this work will lead us into the field of what we might call phenotypic screening, so I'd like to put numbers on this collective behavior. In particular here, I will show you some theory also in collaboration with my colleague Erwin Frey in Munich. Thank you. Thanks, Tom. So now I give you the first very trivial example almost. I'd like to introduce the Porter gene into a cell and see how expression comes up. And this can be done by transfection. And we're interested now at the sort of input-output relation. We put a certain number of plasmids in, see how a gene is expressed. And in time-lapse imaging, what you do is you take images in an automatic way. You measure intensity. You do it nice. You calibrate your intensity into molecular numbers. And then you get single-cell fluorescent tracks that look like this. And what you can see here immediately is these tracks have features and they are different. For example, if you transfect with plasmid DNA, or if you would encode the same GFP on a messenger RNA directly, which you can do nowadays, then the expression looks like the blue curve, which is much more smooth. And the explanation for this is quite simple, because if you transfect with plasmid DNA, your artificial plasmids have to reach the nucleus, and that takes quite a while. In particular, to enter the nucleus is sort of a very late event. While for messenger RNA, all you have to do is bring the messenger RNA into the cytosol, where immediately translation will start. And this very simple biochemical reaction can be written down, translation, and you have two degradation rates, and you can solve this analytically. That's beautiful, because now we can fit our response functions to this equation, and we get for each single cell, we get these three numbers now. So you get distribution functions of translation rates and degradation rates of the particular cell. So EGFP degradation rate is known. It wasn't that exciting, but the messenger RNA degradation time is quite an important number, because much regulation happens on the mRNA level. And in this case, where you try to design mRNA that's more stable, it's a critical number. So what I'm describing here is universal, so each cell has the same genetic code and translation mechanism, so it's no surprise to see it work in different cells. So the initial study, we did it on cells that are moving freely on the surface. It turns out that image processing, if you image over many hours, and this is over sometimes 2 days, all kinds of events happen, like cell division, cells meet each other and merge. So image processing becomes quite complicated, and for this reason we thought it might be some of advantage to sort them on this surface. So various ways of sorting cells, you can attach them or inject them directly, but in our group, Peter Röttgemann and Alicia Pia found a way to let cells organize by themselves. We call it cellular self-organization. The way this works is that you design fibronectin patterns that are surrounded by pegylated areas, which are not as cell adhesive, and then when you seed cells down, you see them, they move, and they start to stick on the islands, and after some time, patterns appear. And then when the grid is filled, you can start some kind of well-defined, time-resolved experiment. So here's a little bit of a chemistry. We do plasma-induced patterning. That means you make stems and put them down to the surface, and then you put plasma in between to make everything hydrophilic. And in these areas, you deposit PEG-PLL, which is a block copolymer with a polylysine that's cationic, sticks to the glass surface, and then you can lift off the stem again and fill the remaining parts, which are the sides here, with whatever protein you like. So this works nicely in our hands. You see here again how cells behave on these surfaces. They are still motile, but they prefer the sides, and they come down and sort of like this one, and now it spreads on the side. Okay, so we studied a little bit on to make this process as efficient as possible. We'd like to wonder which is the best distance you can choose in this case. So we varied the lattice distance, and we measured how many cells are on the correct side. So we call it the filling factor as a function of time. So it fills up as a function of time. The smaller the distance, the faster it fills. And the interstitial cells disappear. So it's all nice, and we try to avoid multiple occupancy. So typically we underfill the chip, so only like 60-70% of sites are filled, and you arrange it in a way that they fill fast, but they do not bridge across the sites. Okay, so this is now the setup. It's an ordinary microscope, some plastic channels, and these microstructured surfaces included here. So now again, same experiment as before, mRNA transfections. The messenger RNA is expressing GFP. You see how the cells are now turned in green, but they are all sitting on the side, so image processing is now very straightforward, and we get all these nice single cell traces. So that allows us to determine the messenger RNA lifetime distribution function. And this is shown here for sort of various different mRNAs that have modified sequences in the beginning. And that's important for a company here that produces therapeutic mRNA that they want to have stabilized. So here's a lifetime prolongation of the mRNA in our cells. Now a second example of a cell response is apoptosis. So here are cells that are dying after exposure to stauroporine, and the death event is indicated by an annexine marker. So whenever the PS is exposed to the outer surface, annexine comes on, is indicated here. There's a late indicator, propylium iodine, that labels the nuclei once the nuclear membrane is ruptured. So again, you extract single cell traces. Now you have two events, and the beauty is now that you can correlate the events within the single cell. So first of all, we can make a distribution function of when cell death occurs. This can, for example, be a function of dose of a toxic substance, like here a nanoparticle, and with increasing dose, cell death occurs earlier. Yeah. But not only you learn something about the timing of apoptosis. You see also these markers being related to each other. So here's the early and the late apoptotic marker. And there's a little time lag that you can resolve nicely now. So typically, annexine comes on 30 minutes before the PI event. But this time difference, here you see it only on average there is a shift. But in fact, this shift is correlated with when the event happens. So in fact, the early cells, there's a clear time difference, and the cells that dilate on the plate, there is a very small time difference that's indicative of somewhat a necrosis process occurring here. So you can extend this concept now for many markers. So this is the correlation that you turn out. So when you extend this principle to many markers, like here is various types of apoptotic events, the lysotracker, cell rocks, this is oxidative stress marker, the mitochondrial membrane, and so forth. After all, this gives us a dynamic signature of events, and apoptosis is a complicated process. There are various pathways, and in order to determine which pathway is followed, this information on the order of internal events is quite important, and you can do this with higher accuracy at the single cell level. Okay. So I skip this. This is just a nice paper by Peter Sorger that I really recommend you to read. It's about the heterogeneity in apoptosis, and that some of this variability comes really from intrinsic noise. That's quite exciting, and of course they also do this type of single cell analysis to access the noise feature in this pathway. I give you now a last example of cell death that is induced now by killer cells. We have two types of cells that are sitting on the islands. We call them the target cells, and these are cancer cells. And then there are killer cells that are targeted towards the leukemia cells by immune labels. We've heard that immune therapy is one of the big achievements in the last years. And you see here how it works. The killer cells attaches to the leukemia cell and it's lysing it. And it's doing it efficiently by this triple antibody that is basically hooking up the effector cell, that's a killer cell here, by connecting it with CD markers that specifically recognize the cancer cell. So in this study, we wanted basically to follow the rates of killing events as a function of concentration of this drug. So again, same kind of setting. We first seed out this target cells, and then in addition, you seed out killer cells together with the antibody, and then you watch how the cells die. So not each event necessarily leads to cell death. Here you see killer cell approaching the leukemia cells. It looks like it's touching, but it does not kill. So it's really something, I would say, not stochastic process, a recognition process that has a certain success rate that you'd like to access here. And what we follow now is simply the fluorescence of PI markers, so the late stage of apoptosis, and you see all these individual killing events here. So this can be done now as a function of the amount of antibody in the sample, and you see that as a function of dose, the killing efficiency increases. There's always some background of unspecific killing, so that's gray is a total killing, and the specific lysis that only occurs in the presence of the antibody is shown here, and that is basically the important sort of drug response function. And what's nice to see as a validation process is that this is the same when we measure it in the classical assay that's established in the mental area. The calcine assay has the same dose response function as the single cell array, so there is no, say, systematic bias here. But the advantage is that time-lapse has now time-resolved capability, so you can study the killing rate at any time. And the killing rate varies, and that's for several reasons. The number of targets disappear, but also the antibodies disappear because they are taken up by endocytosis. So it's not clear that this is like a constant killing. In fact, there is always a peak. So there's an initial increase in killing rate, and then it decreases again. And these are sort of interesting cellular pharmacokinetic insights here that is important to determine the right doses in cancer therapy. Okay, so now I'd like to think a little bit more if the idea to put cells on little sites is clever, or if it has too many artifacts. I call it the price for living in a box. We know that cells on a micropattern are heavily affected by the geometry. The cytoskeleton is arranged. We just heard in the previous talk how stress fibers are formed. Cells can differentiate, they can die, and all kinds of things occur. However, a microarray provides a framework for each cell in a standardized way. So that's a huge advantage. That does each cell sees the same microenvironment. And in this nice work by Manuel Thierry and the company that came out of this, they make standardized frameworks in order to more easily distinguish differences in the cytoskeleton, which you would not be able to detect if they would all crawl around freely. So in that sense, arrays or single-cell microenvironments might play a role if we'd like to put more quantitative numbers to cell behavior, and this is sometimes called phenotypic screening, in particular the hallmarks of cancer that Herbert Devine mentioned as well. Is something that people are interested in. It's not only gene expression, cytotoxicity, it's also the classical phenotypes like morphology, motility, or differentiation that you'd like to follow. Okay, and for this now I'd like to go into motility of, and if you have many motor things and they interact, you see all kinds of patterns, and this is a nice field of active soft matter. We've heard that cells, if they are confined, confinement can induce particular behavior, like a round confinement favors rotation. And this has been known for quite some time and been discovered in the Ingwer group, but recently was quite of interest. Many papers that came out on large arrays, and on the theory that Robert Devine introduced earlier, that explained that cells can rotate in an environment. So we were interested to study this collective behavior, or the onset of this collective behavior in a very small system. Because in large systems there is too much sort of averaging out. We'd like to have a finite size system where we can count each individual cell. Now, so we designed again a microisland, put a couple of cells on there, and yes, you see cells are rotating, but they do not rotate all the time. There are phases where they seem to rotate, but then, as you see here now, they can start to tumble or be disordered, and then they even start to rotate in the opposite direction. So it's a stochastic process, and if you look at the traces, and you can plot here sort of the angular coordinate as a function of time, you see there are times when they all rotate with the same speed, and we would call this a coherent angular motion. So that is a state of collective behavior. While in between, when they are disordered, we call it a disordered state. So we were wondering if we could introduce some type of order parameter to distinguish this as two true states. And Felix, who is here, we tried various approaches for this for order parameters. So the most obvious one is if you take the rotation speed of the collection, you introduce some threshold. And then true, if you now analyze the states that you described as coherent, they show ballistic mean square displacement, so there's direct motion, while all the other states are in a disordered state, as you can also see from mean square displacement. So now what we do is we design surfaces of various size. We make islands of various of increasing size and put increasing number of cells on these islands, and we make sure that the average area per cell is preserved. So we found that important because motility is highly dependent on cell density. So the area per cell in the small island here is the same as in the bigger island with the many cells. And for each of these islands here. Now we determine the times of coherent states. So we ask the questions, if a cell collective rotates, how long does it stay in the rotative mode? And this is described by a survival function that mostly seems to fall off exponentially, so there's a clear survival time of the collective state. And this can be plotted now as a function of number of cells. And it seems to increase, and that's fine. So that's known in many body systems that collective behavior increases with system size. But there is a particular discontinuity. Between 4 and 5 cells, there is a jump. A jump that I didn't want to believe when I saw it the first time, but then Felix came back to me and did more measurements and he said it's real. But moreover, he came up with the right explanation. And the explanation you can only understand when you see how cells are sitting on this island, because there is a certain average spatial distribution. If there are 2, 3, or 4 cells, the cells are distributed like in a flower. They all touch this, the outer circumference. While from 5 on, there is the appearance of a center cell. So a single cell sits in the middle. So that means the geometry now changed, and the geometry is quite important in this case, because what makes cells collectively rotate is that they polarize, and they all polarize in the same direction. Now, the center cell has a problem. It's called, in a frustrated state, it cannot polarize. There is no preferred direction that it can polarize to, and therefore it's unhappy. And this unhappiness, now you can usually see in a simulation that was done in Erwin Freiku. This simulation is based on a POTS model. I will go into this shortly. But you see here the cells dancing as in the experiment. They have a front and a rear. The front is blue. And you see that this cell, whenever it is trapped in the center, happens now, it kind of wants to break out. And for that reason, it destabilizes the coherent state. So what's beautiful is that the theory and the experiment do show the same step at the transition from 4 to 5 cell number. I'm not a theoretician, so I will not go deep into this, but the modeling that I showed you here is based on the POTS model, introduced by Greiner and Rädler earlier. Now, in Erwin's group, they modified this in order to account for designing areas where protrusions happen and where there's retraction. And typically in a POTS model, you dial in when you occupy a new area, and this is given by sort of an effective Hamiltonian. The Hamiltonian includes adhesion and elasticity. But with these components, you would not get collective behavior. It was also pointed out by Herbert Levine. You need coupling between cells, or polarity that is connected. And this, in this model is introduced by a scalar field that the scalar field basically stands for polarization. It gives you front and rear. So these kind of small-scale experiments might help you to understand cell behavior, but it would be nice if you can connect with a single theory, single cell behavior, small-scale behavior, all the way up to collective behavior of cellular sheets. Now, in the simulation, at least, the sheet behavior is reproduced as well. Now, in the experiment, the question is, what about these rotations? Do rotations occur in collective behavior? And yes, we can see every now and then in large systems as well. So I show you just a second. I will show you now experiments where we look at many cells spreading out into a channel. So they have direct direction of flow, but there are hundreds of cells. And you can follow this invasion process by phase contrast microscopy. You can study the flow speed. But you can also analyze this quantitatively using the PIF technology, PIF analysis, particle image velocimetry, that gives you the full vector field of flow. Or you can label the nuclei, and then you see every individual cell moving. Okay, so with this, you basically get the flow behavior. You can see, yes, it flows in one direction. You average the noise out. You see cells flow like in a plug flow. The system has a particular correlation length and a particular persistence time. So it looks a little bit like there is sort of a phenomenological theory of flow, cellular flow, that can be described as diffusion. That means there is a gradient in density and cells tend to flow towards lower density, so there's a transport coefficient, which we call the collective diffusion coefficient. Yeah. At the same time, you could also wonder about the self-diffusion of a single cell, and these coefficients are not the same. And you get something that you have to point out. A single cell is encaged by many others, so, but the entire sheet of cells has much higher motility due to the fact that cells are coupled, so there are bursts with a certain length and time scale. And if you take the correlation length squared over the correlation time in the system, that gives you roughly the collective diffusion coefficient. So beside this, there is also spontaneous formation of swirls that you can detect from the flow field. And Matthias Zorn, who's also in the audience, and analyzed now the vorticity from these vector fields, and these hotspots are basically small little swirls. And what we found is that the number of swirls is a function of density, and this has been analyzed before in the work of David Weitz and Angelini. But here we have a flowing system, and it turned out that in a flowing system there are also swirls, but if you calibrate this to the same density, so here are the resting cells, and here are the flowing cells, and you map them on an axis of cell density, then even though the flowing cells have very low density, they do not have the same amount of vorticity as in a resting confluent layer. So it's a very clear indication that drift seems to suppress the occurrence of vorticity. My simple-minded explanation is that cells like to move, and if they are confined, the only way they can move is in a circle. So there's a certain tendency to form swirls. Now, if you open a box, open one end, and you allow them to drift, then they can basically take all their power to migrate in one direction, so there's no need to form swirls. Okay, but that's too simple-minded. Maybe in theory this comes out more rigorously. Okay, also the vorticity has a much shorter lifetime than the persistence time I've mentioned earlier. So it's only 15 minutes for a swirl. Okay, so now we have some idea of how cells move in a collective. We see them rotating. But having theory, you can now start to predict and design surfaces that should show some predictive behavior. In particular, if you would take the center cell out that I've been talking about before, theory at least predicts you would have infinitely long survival times of the rotational state. That means cells would run forever on a circle like this. So this is the end now. I will just give you some outline of ongoing work within this SFB called nanoagents, where we'd like to design environments to direct cellular reactions. And the first one is a circle here. In this case, we only put one single cell down here, and it's performing basically a one-dimensional random walk. So it moves along this track. Other people have studied linear tracks, but here we can now introduce a barrier. And this barrier is just a little area that is pegulated. And in this case, what we find is the cell is bouncing back from the end. So it's not able to overcome this barrier. It looks like elastic bouncing, but of course this cannot be from all the talks we've heard before. These are photopodia, and the photopodia are sort of running out, but some, and the cell returns. So that's the process here. You see the lamellipodia approaching the gap. It kind of fingers over the gap, but because there is no way to form focal adhesions, these photopods are basically repressed, and the cell eventually forms stochastically a lamellipodia at the opposite end, and it returns. Okay, but however, sometime the cell is able to overcome a barrier. You see it now happening. Here the cell simply runs over it. And in fact, if you make the barrier thinner, it happens more frequently. I call this sometimes the cellular tunneling effect, because the probability to tunnels through the barrier, falls off exponentially with the gap size. Okay. So here's a schematic view on cell migration. We've seen this before, and I'd like to now accommodate this with our analysis here. If you take that behavior on the track and you roll it out, it looks like this. So the states of where it moves, and then in between there again states are resting. You could now apply various types of models. And if you'd like to put numbers to what we call motility here, we have to agree on a model. Yeah? So that some people prefer the persistent random walk model, others like the Levy walk model. Now we prefer the two-state model because, first of all, as you see here, there are clear indications that are in between some resting states, and there are long stretches of running states. But also from a phenomenological point, cells have to have a certain phenotype of running, and then in between there are some shorter phenotypes that seem to be resting states. So again, we do this discrimination in two states, runs and rests. As you see here, there's a resting phase, that's running. And we put now 4 numbers to the cell behavior. We determine sort of the typical run time, the typical resting time, and an average velocity in the running phase. And in case of a barrier, we have a probability for barrier crossing. So these are 4 numbers to describe cell behavior. And in this case, this was a cancer cell line of breast cancer cell that was a mesenchymal phenotype, so it's a cancerous, it's quite motile cell. And people develop drugs to make these cells less motile. One of these drugs is salinomycin. And when you give salinomycin to the system, you see some of the numbers change. Interestingly, what's known is that this salinomycin has anti-migratory effect. So it inhibits migration. Yeah? So you would think that it's going slower, but it does not go slower. Even the runtime is the same. But what it does is it changes the resting times, and it also affects crossing probability. So we hope that we get a little more insight with this model. So here are just some fun movies. We also play with geometries to basically look at the stochasticity of how lamellipodia form by basically making guidance cues that can be reached from 2 sides. And then you see cells are hopping both ways. It's sort of a nice 2-state system in here. And essentially we force a cell to form a lamellipodium at a defined site, which is the end of the guidance cues. And as you can see here, this is a breast cancer cell line. But it looks almost like a keratocytes with very nice formed lamellipodia here. And this is formed every time the cell crosses across this barrier. So this gives us some handle for defined experiments that might link up to what we've heard in the previous talks, modeling of protrusions and so forth. Okay, with this I'd like to thank you for your attention. I skipped the summary and hand over to